Here to speak about the mouse Mesoscale Connectome. They'll be talking about data-driven models of organization and cortical networks. So we've heard about the activity patterns of cells in different regions in response to stimuli, visual stimuli. We've heard about some of the transcriptomically defined cell types in various areas of the brain. And today, we're going to hear in this talk about how some of those areas might be connected. Another important piece of the puzzle. So please welcome Julie Harris, Jennifer Witzel, Cameron Harris, Hannah Choi, and Stefan Mihalis. Welcome.
Okay, can everybody hear me? Okay. Okay, so it's my privilege to introduce our team talk, and I wanted to start by actually introducing the Mouse Connectivity Atlas project to people. This is maybe more of a reintroduction, as this is one of our kind of older but still running projects going on. So the Allen Mouse Brain Connectivity Atlas project was launched initially in 2011. This is when we first started our pipeline, with the goal to generate a comprehensive, directional, and quantitative 3D atlas of long-range neuronal connections. In other words, a connectome. So connectomes, which are the complete wiring descriptions of a brain, or complete wiring diagram of a brain, can be described across multiple scales, as we know internally as well here. We're working on describing connectomes at multiple scales. It can range from looking at a microscale level connectome, where you're mapping inputs and outputs to single cells using techniques like electron microscopy, a project that you heard a lot about at last year's showcase. This also, at the other end of the scale, the macro scale, is something that we're not typically working on here, explicitly at least, but this is a level at which you're generally mapping large fiber tracts between distributed brain areas using techniques like DTI and human imaging. Our Allen Mouse Connectivity Atlas is really a mesoscale connectome, so this sits right in the middle here. And what characterizes the mesoscale connectome is that we're looking at mapping connections between distributed brain areas, but from genetically identified cell populations within those areas. And so this is really the place at which we're generating data for the projects we're going to tell you about today. So as I said, we launched this pipeline in 2011. What we did in the first phase was to map whole brain region-to-region projections. We did this by through generating data from about 500 different tracer experiments. This project really started as a way to help us also set up the initial Mouse Connectivity Atlas pipeline, which was a pipeline that needed to be flexible for future uses. And throughout this particular Atlas project, as I said, we did 500 tracer injections in order to try to get comprehensive brain coverage so that we could map projections across the whole brain, or from regions across the whole brain. I want to briefly take you through the modules of the pipeline, since I think this is really the only time that we're going to talk about kind of how data is generated. Most of the talk today is going to focus on analysis of this data. So in brief, the pipeline is shown here. We start with picking a brain area to target with a viral tracer. AAV, serotype 1, is what we use for all of these experiments. The AAV is injected using stereotaxic coordinates for the most part, although some experiments use different methods of targeting the tracer as well. Each animal receives one injection of a tracer expressing EGFP, either directly expressing EGFP or under control of or in a Cree-dependent manner that I'll briefly touch on again. The brains are imaged after we label the cells and their axons. They're imaged using serial 2-photon tomography. So we map, or we are able to collect and acquire images across the entire brain at high resolution. The nice thing about this, it's black face imaging, so it's very easy to kind of realign all of these images back into a coherent 3D volume, which is really critical for the downstream processing. All of those images undergo some QC steps, manual annotation of the injection site, and then they enter, if they pass this QC, they enter this part, which is our informatics data processing pipeline. And there's really two key steps here that are important. One is the signal detection algorithms, the segmentation of that fluorescence signal, and the second is registration of that image series into our Allen Mouse Common Coordinate Framework, which is a fully annotated 3D reference atlas, which allows us to do this kind of systematic quantification of all these data sets that you'll hear about. Okay, so in this, with the establishment of this pipeline, we published on this first in 2014, mapping connectivity in wild-type mice using the tracers that I mentioned, 500 experiments, and we quantified the projection strength to 600 different targets, about 300 ipsilateral and 300 contralateral. I'll show you just one matrix on the next slide as well from this, and then you'll hear more about how we take this kind of raw data, this experimental matrix, and turn it into a model. So Cam will talk after me about using that data to turn it into a model for some of the subsequent analyses that we want to be able to do. Okay, so the second phase of the Connectivity Atlas project was to try to move more towards kind of this true mesoscale level, which is to map whole brain projections from genetically identified cell classes. And for this purpose, we use Cree driver animals, or Cree driver mice. We ended up with a total of around 2,500 experiments that fall under this particular phase. I also just want to mention that the Connectivity Atlas is almost an umbrella for multiple sub-projects, and we're only going to talk about a few of them today, the ones that are in bold. The most kind of critical, again, for today's talk is the cortical layer-specific projection mapping using Cree driver lines that label projection neurons in different layers and also of different classes. And nicely, you guys have already heard a lot about the different types of cortical projection classes. So we have identified Cree lines that allow us to label both layer, but also IT type, PT type, and CT type lines. So you'll hear the results of that analysis from Hannah later. Okay, so not only we not only wanted to generate this connectome and provide this data as a resource, we wanted to also use it to try to answer some important questions in neuroscience. And the question that we have been focused on, kind of the big question we've been focused on, is this one here: what are the organizational rules and patterns of long-distance connectivity? From the first publication, we did learn some things, some kind of rules or patterns, I guess, about how the brain is organized. And I wanted to just briefly tell you some of the conclusions we had from that particular study. So here's this matrix I was telling you about. Every row here is a different experiment, and every column is a target. And then the colors correspond to the strength of that particular connection. So some of the things we learned included things like connection strength spanned over five orders of magnitude. There was bilateral symmetry, which you can see in the ipsilateral and contralateral side, although there are weaker connections on the contralateral side. There was also a negative correlation of connectivity strength with distance from the injection, and then through several graph theory analyses, we can show that there's a complex organization of this connectivity. There's both clusters as well as hubs. Now, I feel that these are, very interesting features, but it doesn't really seem like a satisfying answer to this big question yet. What are the organizational rules and patterns? So we have continued, of course, to work on answering this question by doing subsequent analyses, and this is what the focus of the rest of our talk today will be. We're looking just at the cortex now for the rest of this talk, and for reasons that hopefully are fairly clear. There's a lot of work going on in the cortex at the Allen Institute, so this helps, and also outside the Allen Institute, since many of our talks have focused on the cortex already. What we're going to tell you about are our explorations into two potential organizational schemes of cortical connectivity. The first one is modularity, and the second one is hierarchy. And so I'm going to pass it to Cam and then Jen to talk about modularity next.
All right. Hopefully you can hear me. So I'm Cameron. I'm a postdoc at the University of Washington in computer science and biology. And I started working on this during my PhD. And I heard about the data set that was being collected here. So this is a visualization of all of the wild type injections into cortex. And you can see that there's good coverage of the cortex as a whole, but you also have all of these gaps. Now that image of the network that Julie showed in the last slide really was visualizing this picture here. So that was showing you for each of these injections kind of which area is lit up. But you really need to process that data before you actually have a connectome, right? Because that's just saying for this particular injection, this spot with this strength, this is what the projection patterns show me. Really, you need a way to summarize all of that data into one matrix that tells what's the connection strength from each point in the brain to every other point in the brain. So we have to deal with the fact that we have varying volumes of injections when you combine all this data. There are gaps between the injection sites. And really, we want to use a method that uses more than just which region was the injection delivered to. So here you can see cortical regions kind of in the top here. Some of them have been cut away in the reference atlas. So just using the regional specification of where you deliver that injection is a kind of a coarse spatial segmentation. Whereas really in the data, we have a 3D picture of the brain and what was fluorescing and how strongly. And so we want to be able to use the actual 3D position data in this method that we're going to build the connectome from. So my approach was to then, instead of building a regional connectivity, actually try to work in the intrinsic 3D coordinates and build a voxel connectivity. So what does that mean? That just means my brain is sliced up into little cubes, which we call voxels. That's like a 3D pixel. And I'm going to try to estimate the projection strength from each of these cubes to every other cube in the brain. Okay, so from every voxel to every other voxel. So that means I need to be able to estimate the connection strengths even where there are gaps, and I don't have data. So that means my mathematical problem is underdetermined, so I'll need some, make some assumptions to be able to solve it. But what we've found is that when you look at two nearby injections, their projection patterns are actually quite similar, which you might expect. Regions have stereotyped connection patterns. So we use that assumption of smoothness in space to simplify the math. So this is how it works. Here you're seeing 4 different injections into the cortex. And you can see the brightest spot is basically where the virus was delivered. Each of those has a little centroid, which I'll call C, and there's a crosshair there. So to be able to predict the connection strength from a voxel where I didn't actually deliver directly any specific injection over here, I'm just going to do a weighted average of the nearby projection patterns. And that's going to be my estimate for the projection strength from that voxel. And we have some more sophisticated mathematical methods for doing this. The movie doesn't work? Okay. All right. Okay, so this is what you get out of it. So I put in all 500 injections. Well, in cortex we have less, but we did it for the whole brain. And I build this matrix that shows you the projection pattern from every voxel in the brain to every other voxel. Visualizing that in 2D, just the cortex, the little red dot is just sweeping through the source voxel, and it's showing you what the estimated projection pattern is for that source voxel. I'll play it again. So you can see as the source voxel moves, the projection pattern changes, and it changes in a way that is more or less smooth. As it moves, you see a blob around it. And then the distal blobs, those are long-range projections estimated by the algorithm. So what's great about this is it's an intrinsically spatial model. It's built in the coordinates of the data, the common coordinate framework. It gives you this sub-regional resolution that you didn't have before. And we've also shown that when you summarize this data back to a regional connection matrix, it improves the regionalized connectivity. So what does that look like? And we estimate how well it does with some cross-validation. So this is what you get when you then take that voxel model and then you integrate it over regions to recreate a regional model. And now Jennifer is going to tell you about the structure here.
All right, so when we look at our connectivity matrix in this form, we can already start to see that there are some regions that are more densely connected, and I've aligned this matrix such that those regions are along the diagonal. But we can also represent this same connectivity matrix in a graph structure where the nodes are the regions and the edges are the weights of the connections between the regions. And so each of these lines here has a weight and a directionality to it. And so you can already start to see, of course, that there are these subregions that are more densely connected to each other. And so what we can do is take an algorithmic approach using a well-defined algorithm to detect these modules of highly connected regions. And I can draw little boxes around them so they pop out even more. And so when we did this at a particular resolution parameter, we came up with 6 different modules in the cortex. And I'm color coding them here, so the same color is on the matrix, the graph, and then I'm showing you in space where these modules are. And so we have one in prefrontal cortex. Then we have a ventrolateral module that's insular cortex, gustatory, and visceral. We have somatomotor regions together and visual regions together. And then there's a medial module that's comprised of retrosplenial cortex and parts of the parietal cortex, and then a temporolateral module. And so it's interesting anatomically that the cortex is organized this way, but we wanted to understand better what this type of organization might mean in terms of function. And so we turned to one particular network that we're very interested in. It's called the default mode network. So this network has been characterized extensively by functional MRI, primarily, also by PET imaging. And these are the parts of the brain that show temporally correlated oscillations when subjects are at rest in an fMRI machine. And these regions also show decreased activity when subjects shift to a task-positive state. And so this is really robust in humans and has been seen in monkeys. The regions involved are prefrontal cortex, posterior cingulate, parts of the parietal lobe, and the medial temporal lobe. And so it's mostly a cortical network. And in rodents, rats and mice, you can see what looks like a structural analog of this particular network when the rodents are lightly anesthetized. Which is characterized by this really strong midline signal, but similarly prefrontal cortex and some posterior lateral regions. So the thing that makes this functional network very interesting to us is that the regions that show this default activity are also regions that are highly vulnerable to pathology and Alzheimer's disease. So when you look at the density of plaques and also of metabolic dysfunction in Alzheimer's patients, these deficits occur in the same regions that make up this default mode network. So we were really interested to see how this particular functional network relates to the structural modules that we identified in the cortical data set. So along with our collaborator, Alessandro Gazzi in Italy, we generated this data set of fMRI data, we made a consensus map basically of the default mode network in mouse, and then we 3D deformably registered this fMRI data to our Allen CCF. And so the red shading here is just showing you the functional imaging data registered to our common coordinate framework at 2 different thresholds. And so then we can take this functional map and overlay it with our structural modules and ask, what is the correspondence between the two? So we quantified the percent of the voxels in each of these regions that were overlapped by the default mode network functional mask. And what we found was that the prefrontal and medial modules had nearly all of their components were part of this functional map, except for the frontal pole. It's the only exception. And then if you look at the other modules, you'll notice that almost none of those regions are part of the functional default mode network, with the exception of the trunk and lower limb areas in somatosensory cortex. But those regions are right here next to the medial module. So this could potentially be a resolution error. So we actually see quite a high correspondence between the functional and structural networks here. So we found that regions in the cortex can be grouped into modules based on their connectivity, and we found these modules using this regionalized voxel model data. And 2 of these modules, the prefrontal and the medial modules, had a really high correspondence with this functionally defined default mode network. And so this correspondence between the functional data and the structural data shows us that this functional network is composed of regions that are actually anatomically very highly connected, more highly connected than they are to the rest of the brain, which maybe starts to give us some hint as to what the function of this wiring pattern is. So I'm going to move on to Hannah.
All right, so another exciting direction we pursued with our crystalline connectivity data to understand the structure of the mouse-brain connectivity is construction of mouse-brain hierarchy. So in primate brain, the hierarchical organization of the cortical regions are very well studied, and also the rules that define the feedback or feed-forwardness of each connections is also very well defined in the primate cortex. So, for example, specifically in primate cortex, the feedback signals tend to originate from the infragranular layer and target the shallow and the deep layer, avoiding the granular layer, layer 4. On the other hand, the feed-forward connections tend to originate originate from supragranular and layer and tend to target granular layer, layer 4. However, in mouse brain, in the mouse brain, we do not really know the actually well-defined hierarchy or the rules that define the feed-forward or feedback connections. So here we ask the questions. So first, we ask the question whether the cortical areas in the mouse brain are actually organized in hierarchy. So are the mouse cortex hierarchical at all? And second, can we actually define the anatomical pattern of connections to define the direction, whether it's feed-forward or feedback? And finally, we ask the question whether and how the thalamocortical and cortico-thalamic connections impact cortical area hierarchy. All right. So in order to do that, we actually looked into the cortical and thalamocortical projection patterns. So based on our tracing experiments in transgenic mice, we discovered diverse layer termination patterns in cortical regions, and we could actually cluster these diverse patterns into 9 different clusters depending on the layer termination pattern in the cortical area. So, for example, in this cluster 1, the connection tend to terminate in the shallow layer, and for example, in cluster 6, the termination happens in both shallow and deep layer. All right. So here is a summary of all different layer termination patterns in the cortical regions. And also, we also included the information from the cortico-thalamic projection patterns. In this case, we looked into the source layer. So basically, whether the connection arises from layer 6 of the cortical area or layer 5 of the cortical area. All right. So we have all different anatomical patterns of layer termination in cortex, but how do we know which pattern corresponds to feed-forward connection and feedback connections? So in order to do that, we actually used an unsupervised method to understand the mapping between the cluster patterns, the layer termination patterns, and the feed-forward and feedbackness of the connections. So here, hierarchy level of each area is defined by roughly average direction of connections from the area minus average direction of the connections to the area. And in order to find this mapping that maps these different layer termination patterns to two of the direction, feed-forward or feedback, we actually search through all the mappings between the layer termination pattern and the direction to find the most consistent hierarchy. So what I meant by most consistent hierarchy is maximizing this global hierarchy score, which measures how consistent the obtained hierarchy is with the directions of individual connections. And by obtaining the maximum global hierarchy score, or in other words, making the self-consistency highest, we can actually cluster these 9 different patterns into 2. So one that corresponds to feed-forward patterns, and the other correspond to feedback patterns. In addition, we have this cortical thalamic source layer mapping. So, it can the connection can arise from either layer 6 or layer 5, and then we group the layer 6 source connections as the feedback patterns, and layer 5 the connections that arise from layer 5 cortical regions feed forward. All right, so we are going to use these 2 different mappings to construct the hierarchy of the mouse brain regions. All right, so remember, first question we asked was whether the mouse brain regions are hierarchically organized at all. So in order to test that, we generate the shuffled data where the source target and the layer termination patterns were randomized and computed the global hierarchy score for this shuffle data and compared that to the global hierarchy score from the mouse brain data. So this is what we see. So here, what you see here is that the global hierarchy score for the shuffle data is always significantly lower than the global hierarchy scores for the mouse brain regions. And since global global hierarchy score measures the self-consistency of the obtained hierarchy. This basically indicates that the mouse brain regions are indeed hierarchically organized. And furthermore, another thing we notice here is that when we include thalamocortical connections and cortical-thalamic connections, in addition to cortical-cortical connections, we see that the global hierarchy score increases as well. So this indicates that the additional data on thalamic connections increases the global hierarchy score. So based on the mapping, the optimal mapping that maximizes the self-consistency of the obtained hierarchy, we can construct this, hierarchy, the hierarchy of the entire brain regions of the mouse brain. All right, so how does this hierarchy of the mouse brain regions relate to the modularity that Jen talked about? So in order to understand the connections, we generated the hierarchy within each module as well as between all the modules in the mouse brain areas. So here is an example of the intramodular hierarchy. So this is from the visual module, and when we constructed the hierarchy within this module, based on the previously explained method, then we see that the primary visual areas is on the bottom of the hierarchy, and other higher visual areas are on the top of the hierarchy. And we can do the same for the intermodule hierarchy. So when we look at the intermodule hierarchy, we see that somatomotor module is on the bottom of the hierarchy, and prefrontal module is on the top of the hierarchy.
So in conclusions, what we looked at was a large amount of data, and we integrated all of it based on a model which allows us to build an voxel-to-voxel connectivity. That is a structure which is really impressive, that it has hundreds of billions of entries characterizing every single location in the brain. How does it connect to every single other location by compiling all our data together? Now, taking this incredibly complex network and regionalizing it, looking how does it map to anatomy, we constructed a connectivity at the level of regions and observed that these connections organize in modules. These modules are important because they are functionally relevant. They map very well to functionally observed modules, and it's very interesting that two of those modules map very well to the default mode network, which is particularly affected in Alzheimer's disease. In the second part of the talk, we have looked at an totally new description, which is going to be a fully mathematical description of hierarchy. Hierarchy has been previously described in cortex, but it has been typically done on a set of observations. Human brain is a fantastic way of building patterns, and Fellman and Van Essen have looked at that, and a lot of other studies looked at those and described, I see this type of pattern, it matches feed forward and feedback. What we did here was using purely unsupervised algorithms to try to discover the existence of these patterns in a purely unbiased way inside of our connectivity. So we described mathematically the hierarchy, a full mathematical description. We found these sets of rules for the hierarchical organization, and we found that including thalamocortical connections increases our knowledge of the hierarchy score. Overall, I would say that both of these schemes describe a cortical organization, that the fact that there is a hierarchy and there is also a set of modularity. So in the end, there is a hierarchy of modules which is present. And in here, we focused very much on describing what are the principles of organization. However, I want to spend 2 slides to describe a lot of other things which have been part of the Mouse Connectivity Atlas, which is the fact that we are interested in discovering a lot of principles, but we also provide a set of foundational data and tools and models which are needed for a lot of our projects. So for example, the data from the Connectivity Atlas has been fundamental in generating the common coordinate framework. Common coordinate framework is a template brain which is fully annotated in 3D, and it's coupled with a 3D reference atlas, and it's a fantastic way of putting together multiple data modalities and data from multiple mice. It has already been mentioned even today in prior talks, both from next generation leaders and from other teams. This data set is also fundamental in order to compare with single cell morphology by giving us all the possible areas in which a particular cell can connect. It is also important in guiding experiments and models in a set of projects which we want to characterize in vivo activity and computations of different areas. For example, this data set is often used to do experiment design, to know where to inject a particular set of viruses, to know where to target the microscope, to target the recordings. It is important as a comparison for the detailed connectivity and retinotopic positions. Just in a talk earlier today, in a team talk, it has been described, it has been used in order to describe the internal structure of LP, and there are other studies from the Allen Institute which have used to do this in different visual areas. We are also working on, and in the future will continue to work on, comparisons of hierarchy position and complexity of neuronal responses between areas, and knowing how to take this set of constraints from biology in order to improve our models and our understanding of the cortex. This is internal, but at the same time, there has been a lot of external use. One of the things which is really in the Allen Institute DNA is the idea of making everything available. So if you want to take a picture of this data and everything which we described today is fully available. The data is very large and is present on the connectivity data set. There is a description of the models on the modeling part, and all the code is available on GitHub for everyone to come and look at it. This has enabled a lot of other labs to do very interesting science. Since the publication of our prior platform paper about 4 years ago, it has almost 800 citations now, so I won't be able to describe all the types of interesting science which it has been used. Just going to mention a few things, which is a lot of people are looking at characterizing via interesting and a lot of good mathematical tools the properties of the connections. It has become one of the default data sets to use a lot of interesting mathematics for connectivity. It has been also used a lot in trying to link structure and function for people who either measure function or try to do simulations of whole brain function. The connectivity data set has been a huge bonanza. It is also a good reference data set when trying to study connection pathologies, and it has been used as a comparative study for elements which link to Alzheimer or autism. And I found it very interesting that there are several data sets which are using very interesting studies which are also have the capacity to link it to learning and even with emotion. Conclusion, I would want to thank our founder for the vision, encouragement, and support. And one of the very important things which is set up here at the Allen Institute is the idea of team science. And I would also want to thank the entire team, which is humongous. A huge number of people have contributed to this. A lot of them are in the room, so thank you very much.
Thanks, everyone. We have time for a few questions.
A couple of quick questions. How do you deal with the areal boundaries? One of your assumptions is this local Gaussian in connectivity, but we know from many species that there are very sharp boundaries, one cell wide between completely different areas, completely different maps, so you would essentially wash all of that away.
Okay, so two answers. In the broader areas, I'm not talking about cortical areas. We actually do wash that away in some sense, but we don't smooth across like thalamus and cortex. So we can build in boundaries, but what turns out, the injections in this data set often cross boundaries as well. So you would expect that to already be kind of smoothed out from the way the data was gathered.
So I would answer that the mathematics allows us to put in boundaries wherever we need.
You know, I'm worried whether the results are actually partly a result of that assumption, and that if you keep, if you are more strict in terms of defining the areal boundaries, and let's say the map boundaries, for example, whether you would see different results.
Yeah, so just in cortex, though, I mean, so we do some cross-validation where we select kind of the width of the smoothing, and it turns out it's small. It's typically it is one width for the whole cortex. So in some areas it'll be over or under, but it's smaller than the size of a whole region.
A second question has to do with this assumption, the asymmetry calculation. You're essentially calculating the asymmetry on the graph to come up with a hierarchy, and it may seem a little bit circular because you're looking for things that are asymmetric in the graph to make the hierarchy, and the concept of hierarchy is also very sensory driven. So I wonder how robust is this? I mean, because your results are a little bit circular. No, you are looking for things that are asymmetrical. Well, of course, you're going to get a hierarchy. See what I'm saying?
So so what do you mean by asymmetry? So is it the number of feed forward versus feed forward? Okay. It does depend on the asymmetry then, because each region is like the hierarchy level of each region is defined by the number of feed forward connections and number of feedback connections it receives and sends. And when you say it's circular, then I don't really understand. Yeah, sure.
Let me try to answer it. So the concept, what we can do is to apply this to a shuffled graph. What we find is the capacity, so theoretically, when we look at what is the possible level of the hierarchy, which would happen in a graph by chance, is going to be very, very low. We are simply going to say, if we were to define a direction, is that direction self-consistent? And that is what a lot of the focus has been on in trying to make sure that that is not going to be happening by chance. So we spent a lot of effort in trying to do the randomization to make sure that that one is not a circular argument.
Just final comment. If you take out the sensory areas and you remain with the remaining of the cortex, you still get a graph that looks...
Talk about the scores for each module?
Yep.
Almost?
Yep.
I don't know. Or just say we will do that.
Well, so you're saying that, okay, so without the sensory module, so we actually did the intramodule hierarchy, right? So when you actually look at the module, like each module, and then try to construct a hierarchy based on the same algorithm, then we get the reasonable hierarchy as well. So whether it's sensory or not. I mean, one more thing. So, like within the sensory module, the hierarchy score is definitely higher. It's more hierarchical compared to non-sensory module.
Down here.
Your inter-module hierarchy had the somatomotor module at the bottom of the hierarchy. Was that predicted by previous studies? Were you surprised by that?
Yeah, I was surprised by that. I mean, I think in some ways we expected it to be visual because so much is known about that. But I mean, even though I think it's because, in some ways it might be because of the way our CCF is parcellated too, I have to say. So we have, in the somatomotor module, there are a lot of primary somatosensory cortical areas. So each of the subdivisions of primary somatosensory cortex is delineated, so mouth, nose, upper limb, lower limb, whereas primary visual cortex, there's one, right? And then many higher order visual areas. So I think that when we look at the intermodule hierarchy, that might be one of the reasons it's pulling the somatomotor one down. Yeah.
Okay, thank you.